It's welcome. So let's start. So this afternoon, I would like to go to the basic. Huh? So of course this is one of the. It's not a matter of you don't know or you don't have the skill to do it. Of course, all the science that you learn during your graduation, if you don't use it, you forget some of the concepts. And sometimes when you are in very sensitive discussion within the Codex Alimentarius, and you are talking about percentiles, about mean, about median, ADI, TDI, you remember IFFT. And sometimes I was listening to the discussion between the delegates, and I see there's some confusion about some of the basic concepts. And when you ask people about what is percentile, you are always a little bit lost. So just I put an example here. You have this graphic. What is the name of this graphic? What do you call it? Huh? Histogram. Yes, this is an histogram. Huh? You did an histogram by hand during your and the high. So you are counting. So each block represents 10 individuals, 10 values. So that means here that this is the amount of I don't remember which food it is, salads per day, eaten by person per day, and you see 10 people, they are eating around 10 grams per day. Okay. Another 10, around 20, and so on and so on. And you see the majority, high number of they are around 50. So it's a way to show the distribution of the daily consumption of salads. So it's a representation. So what's this graph telling you when you look at it? Yeah, almost 50. So no, well, no, it's not a question. No, you see, but you are very technical here. I don't want to be practical. So the data is here. The median, the normal are tools that you are using to describe things, to do statistics. But at the end, when you are doing statistics, is to do interpretation of the data and be able to use the data. So it's showing me that there is variability of the consumption between 10 and 90, and majority of the people are around 50. This is, I don't need to put any technical words. This is the function of this graph. Now, if I want to use those data to calculate, for example, the mean, so what I do, I put the values 10, 20, 30, etc. And I put the frequency of each value. I multiply them. I sum them and divide by 310, and I calculate the mean. This is the average. Easy. You did this calculation. Sure, you did it dozens of times. Okay, so this is the median. So this is okay. Median, the mean, of course. Sorry, the mean. I put it in Spanish. Media is the mean in Spanish. Huh? Sorry. Is it useful as statistics? No. If I want to use it for management, it's not very useful. So what is something that can be more useful for me? No. Okay. So, for example, if it is about nutrition, let's salads is a source of fiber. And you want to put recommendation. recommendation saying it will be healthier if you eat at least 50 or 60 grams of salad per day. Okay. So which statistics or indicator you need to know? Now, how much people, what is the proportion of people eating, reaching this objective, and how people are less than this mountain, this amount. This is much useful than the theoretical statistics where no one understands them. So if you said, okay, now look, I have only 20% of my population there at the objective or more than the objective. Okay, that means that you have an effort to reach 80% of the population to improve. For the manager, this is what you need. Huh? That's why we need those percentile, because you can ask yourself differently. What is the 90% of the population? What is the value? 90% population are eating a certain amount of food or not. You see, so this is the percentiles, but a lot of people, they don't understand that, but it's better. I don't like those histogram, but we are used to see as, oh, it's normal distribution. No, something better than this is what we call it, the cumulative distribution. So I will show you how we do cumulative distribution. So it's not complicated at all. There's different steps. You know the difference between complex and complicated. No? No. That means there's why it is complicated, because you have different steps, but each step is very simple to understand. So you have the values, you put them in order, and you have for each value the frequency of each value. Okay. Now, because when you do the histogram, I put here the center of each class. Huh? So the maximum of each class is, the first class is 15. It's between 0 and 15. The second is between 15 and 25. So I put for each one the upper limit 15. What does it mean here? That means that 10 people declare 15 grams or less. Okay. So after that, I move to 25. What I do? I add 10 and 10 plus 10, 20. That means I have 20 people declared consuming 25 grams or less. You got it? And you move until we reach the total number. Now, if I use this number 10, 20 in those one, so this 10 is coming from this block, 10. The second center 10 is coming, I have 10. Here I have 1, 2, 3, 30. Here I 1, 2, 3, 4, 5, 50, and so on and so on. And the last one, I have 20 here and 10. Okay, sorry, I didn't go to all the steps. Now, because it is here, it's between 0 and 15. So those 10 declared 15 or less. Okay. Here, these 10 are between 15 and 25. Because here I put the center of each class. Okay, now, this 10 is exactly the same. 20, I accumulate 10 plus 10, plus 10. Voila, 20. And after that, to 20, I will add the 30. I will have 50. Cumulative. Once I finish that, I divide by the total, because here, 310 represent 100% of my sample. So 100% declares less or equal to 95 grams per day, which is the maximum I have here. Perfect. Okay. Now, so, and 10 represents 3%, 20 represents 6%, and so on. So instead of having this histogram, I will represent here the cumulative distribution. And you see it in different publications. So I prefer using those cumulative distribution rather than this graph, because it's more informative for the manager. Because from this distribution I develop, I can say is 55. How many people eating less or equal than 55? I can know how many people are eating more than 55. And this is the information that we need. For example, if you are looking for the maximum level, you put the maximum level, certain thresholds. They say for aflatoxin, 5 microgram per kilo. So what you want to know, what is the proportion of a product that exceeds this amount? So it will be the complement of this distribution. So if you have this cumulative distribution, it's much easier to use. And I don't care if it is normal or log normal or whatever it is, I don't care. And this is very useful. So I think this cumulative distribution is the more important distribution that we are using when we are dealing with risk assessment. Because risk assessment is always about threshold and how much succeed, how much do not succeed. Compliance, the same thing. When you are doing compliance, what you want to know, how many company comply, what is the proportion of products with compliance and without compliance. So this is so when we say percentile, so if I say, what is the 90 percentile here? If you look at, no, the value. Okay, so the more here, I have almost 90%, is okay. So I go here, and it is here, 75. Yeah, what you are, the percentile is the probability 90%, and the percentile is the value that reach 90%. So if I look at 95% or something here, 85. If I'm looking at 30, so of course, because the data are not well presented, but you have software. Now, if you put the data in a software, you put, I need 90%, 90%, I need 75 percentile. It will give you directly the figure. Okay, so I spent 5 minutes on that because if you don't understand the concept of percentiles, we cannot understand the next step. So back to the basic. But it's something that you need to have it in mind, and you are using percentile daily. This is the probability to exceed. So like we said, you can, when you look at the bottle, you have at the half empty or the half full. So you can look at what is the proportion less than a certain value, or you can look at the proportion more than a certain value. Okay. So if you can promote the use of cumulative distribution, thank you for that, because it's very useful. Huh? It's more useful than the median, the mean, and okay. So another concept here. So when we are working on with the analytical tools, so you have this value, possible value of the concentration that you estimate from a sample. So we classify, we have LOD, LOQ, LOD limit of detection, LOQ limit of quantification. Okay, and here we call them quantified. Okay, and here we put them non-quantified, less than LOQ. This is the only information that we know. What we know, they are between LOQ and LOD, because the determination of the value is not enough good, is not accurate between this interval. And you have non-detected, you have 0, but it's not real 0. We don't know. So you put non-detected. Now, you have data with figures and data without figures, because when you said is less than LOQ or less than LOD, you cannot use them as numerical value. So what you can do, you have different options. So the lower band, that means all the non-quantified, you put them at LOD. All the non-detected, you put them at 0. The upper bound, you said, no, I cannot take the risk for the consumer. I'm more conservative. Huh? So the non-detected, we put them at LOD. And the non-quantified, you put them at LOQs. Yeah. So you substitute the value. It's easy. And we calculate, we have the upper bound and lower bound. So this is important when you want to use those data for exposure assessment, because you want to estimate the average concentration. So the fact that you do UB and LB is you create a bias. Because the true mean needs to use the real number here, but you don't have you don't have the real number, so you substitute. So you calculate the mean with UB and you mean with LB, and you look at the difference between them. If there is no big difference, you can say there is uncertainty, but it's not too big. For example, if you calculate the mean with UB, and you find, for example, 3.5, and with LB, you found 2.9. Is it a big deal? Yeah, not a big deal for the concentration. But you need to do the exposure, and you see the difference between the exposure. So sometimes if your method is not enough accurate, we can find the situation when you calculate UB, you can have people exceeding the thresholds or not, depending using UB or LB. No, I think because they didn't give you that. No, because they provide you only the LOQ, so you don't know. Hmm? Yeah, yeah, yeah.
The validation data, the link is between there are the difference. At least there are 10 times more, 6 times different.
6 times different. So if your LQ is very low, for example, 002, 002, dividing 002 by 6, it's not a big deal. The impact on the of the mean will not be huge. This is a common sense. It's not a rule. You try different way of calculation and look at the impact, and you decide after that if you change. If you use one option, do the final conclusion will be modified or not? Yeah. Yeah. Yes. Yeah. Usually we use the upper bound approach. Yeah, they have worst case scenario, but when you are, we always use do the calculation for both, because if with LB we don't see a problem for exposure, and you see the problem only because you are using UB, we said we are in uncertainty situation. We cannot conclude. Okay. Usually we need to be transparent and say there's possibility, but it can be due to the fact we didn't use the right, the accurate analytical tool. Yeah, yeah, we show. So so you are safe if the conclusion, the final conclusion, will if it is the same, will not change the conclusion. So the impact of UVLB on the conclusion, nothing. So you can continue your process. But if the fact you are using UV, you create the problem, we don't know, it is artifact because we move the value from somewhere in between to LQ. So this is will depend on the range of this, depend on the quality of the methods. Exactly. And usually when we start to plan to design total dye study, when you are looking at different contaminants, we do some simulation to see if we can be in those situations, and we choose the analytical tool that will not put them in that such situation. So we do some calculation before. So depending, for example, depending on the ADI or TDI, if the ADI or TDI is very low, you need to have more accurate system. So if the ADI, TDI are in milligram, not in microgram or nanogram, you see. So it's, yeah, you have to look at it and predict that in advance. Not you need to be proactive. And we did it in France for each TDS. We have a call to ask laboratory in very competitive way, which of you will be able to reduce the LOQ to a certain level, because they can adapt, because you are not here in official inspection. When you are doing exposure assessment, you don't need to have ISO methods, because you are not going to the trial because you are doing exposure assessment. There's no trade issue or something. So it's mainly sort of scientific. So you can work for some substance with some university that are developing new tool that give you more lower limit of, and you know, it's this method is cost is more expensive, but it will not be used in routine, but especially for the study. Yeah, for exposure assessment, it's very useful. Okay.
If we need to study the exposure assessment with current capability or current analytical methods which we have for inspection or for the regulatory lab, it will be very difficult now based on the approach of the risk based and based on the approach for exposure, because all of the laboratories or all of the available analytical methods, it's built to be fit for purpose. Yeah.
Type of laboratory. You have the commercial laboratory and you have the reference laboratory. And usually when you are doing those exposure assessments, you will work more with the reference laboratory rather than with the commercial laboratory. So yeah. So, but it is a special request. It is a special request for the study. You ask them to adapt the method, and you know that you have the reference that this method can be adapted. Huh? Yeah. Mm-hmm. Yeah. No, but,
years ago in Egypt, we have the symptoms.
mhm.
Differentiate between
Yeah, and because the government, they don't have enough resources to maintain those. I know I know the situation. Yeah, you can use academia and yeah, uh, and we don't need to have reference laboratory everywhere in each country. So you can do it at regional level. So instead of doing one everything in each country, you say, okay, we know that in Qatar, the university are very good. I don't know which method, HP, SPS. So they have the experience. Okay, you audit them and you approve them as a reference laboratory for the region. Another in Egypt, another in. So you don't need to have them everywhere in each country. So this is a regional strategy that need to be implemented because we will not have the resources to have a reference laboratory in all the countries. It's impossible. And this is what we still have in France, in Europe. France still having national reference laboratory, and some of the national reference laboratory are EU reference laboratory. And I think with the time, with the resources, those national reference laboratory will disappear. Huh? And we'll use only European laboratory. Okay, so here we are. Let's move with the, okay, so I would like to switch here to, yeah. Yes. Oui. Yeah, I forgot to say that. No. No, just for the completeness, I want to show you that you have in between. No, you some because no, when you publish with the journal, sometimes they are we are tired with all those new editors in the journal. They become no, you look what happened for the newspaper in general. Oh, you need to make it attractive for the readers. The same thing we are doing for the science. Make it simple, make it attractive. So it's crazy. We are in crazy time. Crazy time. So you don't see. So people they do it in their lab, but they don't communicate it because at the end they communicate what is needed. Okay, so we simplify but there is other approaches. More complicated approaches, quantitative approach when you have enough data. Because when you are in trouble, you're in trouble when you have the majority of your data, censored data, less than LOQ or less than LOD. So when you have enough quantified data, we can use the modeling approach. You can do modeling approach, and you can take different assumptions about the distribution and do it in more rational way. So you can, there's softwares, there's a program that help you to do it. So it's not the objective of this, but if you are interested, there's a technical approach that are useful, and there is a report, EFSA report, describing the how to deal with LQ, LOD, and they cite the maximum likelihood approach, Bayesian approach that can be used. So there is a consensus among scientists. There's when you have a minimum number of quantified, we can use those approaches. And there's one team in which country? Norway, yeah. They develop Bayesian approach for exposure assessment, and you can put your data with LOQ, LOD, and doing the right calculation. Finland. Yeah, yes. No, it's easy. They are doing open software. I can add some the link, the paper and the website where you can use it. Okay, now let me introduce, try to summarize, and we go to the example calculation. So, so we are in our process in Codex Alimentarius. We have question about cadmium, about aflatoxin, and we do start to do risk assessments. When we start to do risk assessment, we go through the full process, hazard identification and characterization. We provide ADI or TDI or benchmark dose. And we do exposure assessments, and we end up with the risk assessment. So what are the conclusions of risk assessment? It can be, okay, this is the effect, carcinogenic effect, can be reproduction effect. We provide the TDI, and we do the exposure assessment, and we can see that some of the conclusion we said situation of concern. We never say risk when we see that there is a potential that a certain proportion of population can exceed the TDI. You want to leave? Huh? You want to leave? Yeah. It's okay. We have okay. Yeah. Okay, we'll organize. No, I think this is a starter. So with Samuel, we will try to organize. Yeah, thank you. Yeah. Thank you for your active participation, and see you next time. Thank you. Yeah. Okay. So so we say when we have a proportion possible, we have with the calculation that a certain part of the population can exceed TDI. So we said it's a situation of concern. Does that mean there is a risk because you have some margin of exposure? So from that we can decide. We go to risk management. And what will be the decision of risk management here? Do the chemical, the question of a risk manager, do the chemical of public concern? Yes, no. If yes, if no, nothing to do. If yes, how to control the exposure to the chemical? How we can control the exposure? If the exposure is of concern, how we can reduce the exposure? So we can reduce the amount of the concentration, or if we cannot, or reduce the consumption. we have two options. Huh? So no, for example, for some products, we say for children at this certain age, avoid this sort of huh? body weight. But to increase body weight, you have to eat more before. Okay, so uh, here, how you usually to we try to reduce the occurrence. To reduce the occurrence, you develop code of practice and you propose management, good manufacturing, good for aflatoxin harvesting, storage, and so on. So we know how to reduce the concentration. You implement, so 2, 3, you implement. When you implement, you need to analyze monitoring data. You need to go to the monitoring. We have food contamination data, food consumption data, because you can, that depends what you recommend. Sampling and compliance. So here, the maximum level could be interesting. Why the maximum level could be interesting? Because you can put a certain target. Because maximum level without code of practice doesn't make sense. If you put maximum level without code of practice, that means you are just rejecting. Okay. Okay. And here, so you can decide from that with the implementation that you need to put certain standards, maximum level, for example. And here, when you are doing, you have decision making. So where to put the maximum level? How to do the maximum level? And this is here a different way to do it for residues and for contaminants. Alara, wait, but yeah, it's not, it's Alara, but not Alara. Yeah, exactly. And this is the trade-off between how much I can accept to reject. Oh, merci beaucoup, c'est gentil. Yeah, okay. So here, so once you have, you know the situation. Yeah, once you know the situation, so you decide to propose maximum level or not. And the maximum level, how to decide it is about the trade-off. Huh? Trade-off, how much you reduce the exposure and how much you accept as a rejection rate. And after that, you put enforcement and monitoring and so on, and the cycle, you continue the cycle. Okay, so this is the approach of, so you cannot, for example, once you decide for risk management, if you don't have those data about monitoring, you cannot set up maximum level. So sometimes we forget 3. We jump directly to 4. Yeah, because I can put it before here if you want. But it's yeah, but that depends if it is a new contaminant or not. Huh? It's dynamic, so it's not a real order. Huh? So, but this is what we need to end up with maximum levels. You need risk assessment, risk management decision about the need to do something or not, having the monitoring data, important, and making the decision about the level where to put the standard, the maximum levels. Okay. And communicate, of course. You need to communicate about all the to explain the process because sometimes it's very strange. So, just few examples. Un quart d'heure, on s'arrête. Okay, okay, okay. I need to. Can you switch? Yes, now it's. Okay, so I will show you the example first. I need to. Okay. I need to increase. So I'm using not your data because with 11 data, it will not be possible. So let me know. I'm trying to increase the size and let me know when it is enough. You can see. So we have this survey. I take out the names. I don't need the names. Huh? We have the gender, we have the age, the body weight. We don't need this one because I will hide it. And we asked people, so this is eating a few, almost nothing. We have the 7 days and for each person. So you can see here, for example, this person, you see the variability. Some days they eat more, other days they eat less. So I have here 300 participants. And here, what I am doing here, so I'm calculating the average consumption per day divided by the body weight. So what you have here, this is the unit of consumption that we are using in general. We don't express the absolute amount of food, but number of gram of bread per kilogram. Okay, we standardize. So this is, you have the distribution. You have the distribution of the consumption. Okay, so you can. When I put it, oh, I hide it. So just. Sorry for that. Basically. Oh, where is the graph? Here, I put it here. So just here to show you the distribution I present here, the average, yeah, I will show it later. I will show it later. Now, so something. So I will take this one here. So remember, yesterday I showed you the results of the mean of the concentration of OTA in two regions in Morocco, littoral and continental. And remember, it was 132. So I was not using this probabilistic approach, what just using very simple way, because it would need more time to explain the approach, the probabilistic approach. So here I put the bread consumption, the calculation I show you. We did the calculation for the total person who participate to the survey. And I have here the average bread consumption in gram per kilogram of body weight. Here we have the median. Okay. Here you have the 75 percentile. What does this mean? Equal or less than 4.16, and 25% more than 4.16. And I do it for 80 P80, P90, 90 percentile, and 95 percentile. So this is, I will show you later the other graph. So sometimes you are not able to go beyond 90% or beyond 75%, because to be able to estimate percentile, high percentiles, you need high number of observation. There is a statistical validation for that. So you need to be careful, not everything. So there is tools that give you the confidence intervals to give you an idea if it is, because you will end up with a huge confidence interval because you don't have enough participants. So you need to be careful. You cannot calculate percentile with small number. So for example, this is sometimes stupid things that you found in some publication. They have 30 participants and they calculate 90% or 30%. No. Usually we explain to the student, if you don't have less than 100, don't try to start to calculate percentage. You want to say something?
Question when we have is about our distribution. We have a distribution, and sometimes when we analyze the data, we will find some points that are, we can call them
extreme value.
Yeah.
Outlier.
Yeah.
Yeah. Outlier. I will show you.
Aberration.
Okay. Aberrant. Outlier. So we call them outliers.
How to manage these data?
Yeah.
I know that we have the possibility to correct our. Wait.
I have the example here.
Thank you.
You will decide. So look. So here, what I did here, I remember that the TDI is expressed in weekly day. You remember yesterday it was 100. Okay, let's continue the process. So for the mean, for the median, and for each percentile, what I did, I calculate the exposure by multiplying by the 1.32 and by 7 days to end up with week instead of day. So you have those estimation. So what the figures tell you now? What will be your conclusion about the exposure here? So we have in average 34, median 50% of population less or equal than 50, P90 is 83, and P95 is more than 100. So what will be the conclusion of the risk for the risk manager? Remember the same issue. No, you want to take the microphone? The same issue. I don't want you to do technical interpretation. Look at the data. I said, what is the meaning for the risk? Okay, I see there's difference in between. Recall, what is the objective? What is the objective? We estimate. Yes, we have we calculate the exposure with the 100. Okay. No. Okay. So if you answer to me very quickly, I will say it in French. Tu es le médecin. Je dis, c'est grave, docteur, ou pas? No, how about next step? Is it bad or not? I want this. And after that, we can go in details to understand.
Because the P95 exceed the
Yeah.
reference value.
So we have a certain proportion of the population, 10% exceeding. But before that, wait a moment. Before that, look at the 75%, 38, P80, 49. You can say we are far away. But wait, we did the calculation only for bread. Hey. Only for bread. Other sources can contribute.
The bottom line is you need to work at decreasing exposure.
Yeah, exactly.
That's it.
Now, you have some technical question because always the problem when you present those scientific, it is scientific approach or not, whatever it is. You did serious work, and you will have always people who want to put some doubts about your results. So you said outliers. Okay, outliers. So because, yeah, you count. So I show you here. What you have here is the distribution, the cumulative distribution of the exposure. So I put it on log scale because it's nicer. And the confidence interval. So you see here the, but now if I go to the details of the calculation, which is the exposure here, give me 2 seconds to set up. So if I go to the highly exposed people, because it said, yeah, your estimation or your survey about bread was not good, and you have high consumer, high eaters. So I can see here that I have some of them. Yeah. But they are not representing more than 5. Is 1, 2, 3, 4, 5, 6, 7. 7 out of 300. We can't say by mistake. They said they are eating 4 kilo of bread per day. Well, okay. So we can clean a little bit, but if it's not change. So that's so those details, be careful those details, because you can lose the trust on your data, on your work, because someone who high and people who they will not understand that will not change the so clean your data before you present them. Huh? So be careful with the communication. Make your case strong.
Cleaning like anything that skews the data, trying to clean it. I mean, I think doing the analysis with the data unskewed,
Yes,
and skewed both. So
yeah, the same thing that we did with the UV-LB. We do the calculation UV-LB, and you see what is the impact on the conclusion. The same thing here. We can take out the outlier, and with and without outlier, and look if there is difference. And we say, so in this way you are safe. Because it's very, very, very complicated sometimes. It's someone with a strong voice saying, "Oh, you didn't, you took a high consumer, that's why you are." And try after that to to explain. Huh? So a good question. So this is it's a simple way to do the calculation, and so the complication is to get the data, not to do the calculation, and that's why this workshop is mainly to encourage you for data collection, have the data of good quality and so on. Okay. Are you ready for a maximum level or? Okay, let's go. It's a simple example. So we have the basic to understand. So we know the percentiles now. Well, okay, so. Give me 1 second. Okay, I have to. First, you have you can have a decision tree in different step. Remember this what I show you here. This you do the risk assessment, and as we did here, we said risk manager. The question of the risk manager: do the chemical of public concern? Yes, no. Here, what will be your answer with OTA, with breath? Yes, yes. Okay. How to control the exposure to the chemical? So we have two choices. We said you play.
I mean, again, here I would, to be honest, I'm very codex bound, right? Like I would go back to the general guidelines for
yes, yeah.
management of contaminants in food, which essentially tells me that I need to apply the ALARA principle. That's essentially what it tells. And also it says the main thing is that we need to look at preventing the introduction of the hazard as much as possible. So as early on as possible to intervene. So it means that if we have any producers, if we have at various levels, so trying even to do a quick analysis of what are the sources of the hazard. Like if we are a strictly an importing country, the answer is very quickly done. I will have to check my imports and I will have to manage that. But if we have like a mix, we are producing, we have issues of storage, particularly OTA. OTA is a storage and transport, toxin. So it means that I may have some handling issues that I need to look at. So I would identify that as part of the bucket of things to intervene. So most of my interventions need to be actually looked at from the standpoint of intervening within the industry. But while I'm doing that, I would be considering two things. Do I need to develop sentinel levels for industry to look at? And when I say sentinel levels, that's actually a maximum level, enforceable or not, depending. Some sentinel levels can be non-enforceable if they are benchmarks for the industry. So essentially, and these will be based on how we study their manufacturing practice. But then I will make a determination as to, or we will make a determination as to whether an ML is a warranted or a highly suggested risk management strategy. Because again, the idea is not to have an ML if I don't need an ML, because otherwise we will be managing things with
that's
MLs.
why we start the discussion. First, we need to look at the origin of contamination. We need to develop code of practice. First is you need to have code of practice.
No, I'm just actually answering the fact whether you need an ML. And again, I mean, I'm sharing with you here the Canadian perspective, which is very much the codex perspective. We have two situations where we consider an ML is warranted. One, when essentially we know that that commodity is a major source of exposure to the contaminant. So if it is a major source, major contributor to the exposure, by putting an ML, I may be systematically driving the levels down, therefore reducing the levels down. And that an ML would be a good one. The second thing is also, it might facilitate enforcement. So because I don't want to be doing risk assessments every time I have an incident, basically I set a level and it becomes a systematic management of the situation and a more predictable environment. Those are,
it's we are saying almost the same
thing. I think we're saying the same thing.
And we said that it is very important that before the ML, is a target for the industry. It's very important, and you cannot put it very low immediately. You can put it in a successive way. You implement, increment, step by step, and and before you set, because that depends because this step 3 that put it here, that we need to have monitoring data, it can be before risk assessment or after the risk assessment, depending if the problem is new or not in the country. Okay. So before starting to look at decision about level to put maximum level or not, you need to have data, good monitoring data to understand the situation. And this is what we are doing in, for example, in Codex Committee. They want to update the maximum level after the implementation of code of practice. They said we need to have new data. So you need to have new data to have an idea about the current practice. Do we change this situation or not? So this is one of the messages. You cannot start putting a maximum level without having those monitoring data. They are very important, unless you decide to do it in unilateral way and said, I want to protect the consumer. I don't care about the capacity of the industry to reach this target or not, which is not the right way to do it.
If you look just
Yes.
about the action level, sometimes we have to define
This is different.
action level and what the
Yeah,
differences there.
this is mainly used for LEDs for children. It's we call it the internal exposure assessments. It's not what you are eating, but mainly about the because lead is a little bit complicated because there's now we are no more threshold. We are putting down and down. No more threshold. Yeah, action. Yeah, usually the action is lower than the maximum. That depends. But I think when you don't have maximum level, you can create that action level to do. Yeah. Yeah, Sentinel.
Found it it's among all the recommendation of EU in their regulation, where we don't have the threshold levels. They request to establish action level to know if the countries are establishing monitoring and to see if they have some issue about contaminants. So
yeah. Now, I think, yeah, one of the we can put it different way. So sometimes we have this issue with the dioxin in Europe and in France in dairy, for example. It's more than 30 years ago. They don't want to invest data on monitoring. So a certain way is if you put a limit, you push them to do the monitoring. So you put implementation, say, yeah, if you don't provide us data, we need to do something. We put something arbitrary. If you're not happy, provide us the data and we can discuss later. So it's another way to communicate with the industry to push them to do something.
I understand that when we want to establish action level, that we have first of all we have to do exposure assessments and to see if by defining this action level we are exceeding the exposure and we have to take some management measures to mitigate risk. That's what our understanding of this situation.
No, but that they are they are possible option. But the difference with the maximum level, it has official. It become official. It will be used for the trades. It will have another significance, and we end up with compliance, non-compliance. And don't forget that, compliance, you need to have the sampling frame, the sampling plan in addition to that. Okay, let's give you this example. Okay, I try to increase just to show you the data. So those data are new, coming from Africa about aflatoxin. So there's a lot. Yeah, I will increase. Yeah, sure. Okay, I will. Is it enough? Or a little bit? Okay. So those data was you have different foods. Rashid. So if you want to have, yeah. Okay. It's not working. Yeah. Okay, so you see that you have different products. You have, okay, so just for the example, so you have different aflatoxin here. We calculate the total aflatoxin with LB and ULB. And no, I saw, no, I didn't try. It's not the right one. Sorry, I I didn't open the right file. Sorry for that. Aflatoxin. Oh, why I don't have it? Okay. I will open it from a mixing. Give me more seconds. I will give you the send you the all the data. Yeah. No. Yes, it's here. It's here. Okay. So we have this. We have those data. Okay. Now, if we look at the of course, when I did the calculation, I need to use the UB, but for maximum level, we use them just to correct, but because we are not calculating the mean of the contamination, but mainly we are looking at the rejection rates. So we have here 406 observation data points. So we have, I calculate the average of total aflatoxin. So you see from LB is almost 52, 52, UB almost 52. So you see the impact of UB-LB is very low. So it's no big deal here. I calculate the P50 and for LB and UB, these small differences. But when we go to the high percentage, we no more see the difference. And P55, we have 249 and maximum, yeah, a huge maximum. So there is some situation when we have, I need to increase the size because it's not good. Let me, because I open it with the driver and we don't have the same possibility. Okay, much better. Okay, so we don't need all those digitals. Just so how in Codex Committee they are doing that? It's not doing that in very complicated technical way. So easy. It's not complicated. So here I put possible maximum level. Possible maximum level for 10, 15, or no ML. Okay. If there is no ML, 0% of rejection. Very easy. And here I ask I have the data, I have an equation, and then calculate for me. If I put it at 15, it will be 25%, at 10, 28%, and 4 at 36%. So those data are coming from African markets. So to understand the context. They are not export products. Huh? High rejections. So it's plausible to see this kind. Yeah, knowing the climate, knowing the humidity, the storage condition. Okay. Now you can I do a very easy calculation. No? It's not very complicated situation. I look at the mean of the contamination, because at the end of the day, the mean of contamination, you use it for the exposure. Okay, so I put as a reference the more stringent maximum level at 4. Okay, and I say if I move to 10, I will multiply by almost 2 the average concentration. If we go to 15, 273, and no ml 80. Okay. So what is I recalculate the average? I exclude from the database all the value that are exceeding the 4, 10, or 15. It's very easy calculation. And this is the maximum of reduction, because I assume here that all the batches are sampled and all the analytical tools are working well. I don't have errors in the determination of the concentration. Okay. So having those information, what will be your proposal for maximum level? Or if you are representing those African countries and you are in negotiation of new maximum level, what will be your your position? Did you understand all of the figure before we move on? No, yeah, they mean ratio. Yeah, ratio. Okay. I will, okay. Yeah. So here, what I did here, from the database, I exclude the 36% that exceeds 4. I clean all compliance. I calculate this mean. Okay. So if I tolerate until 10, so you see, you increase the mean, 1, 2. If I tolerate until 15, I will increase by 1, 7. And here, 51, you explode. So just to forget that, I put this at 1, the reference. And then if I move my maximum level from 4 to 10, I will multiply by 2, the ratio. This is this value divided by this one. No, it's the way, sorry, this way to represent. And here almost by 3, and here almost by 18. So you exposure the potential exposure by 80 times. Okay, thank you. Okay, thank you for the question. Because sometimes we go faster. It's my reference. It's my reference. So this is the costs if you move for less stringent maximum level. Right. I can do it the other way. Yeah, yeah, but it will be yeah. So you have those. Think about it, and how will you participate to the negotiation? It's difficult for this country. It's tough situation. Yeah. Yeah, and this is the problem here. So you don't have a huge difference, because that means that they have some outliers or some what they put in their data. You remember our discussion this morning. you see. So, but if they submit those data, they cannot clean them after that. And this is That's why the objective here is not to go through the full process, but just to show you the situation when they can be in trouble because they don't care about the sampling frame, representativeness, and so on. Because if they look at them carefully, perhaps when they sample, those situations can occur because they are extreme situation of bad practice that can be manageable. So when you are doing maximum level, should you include but you see, yeah, but you still have a high number at 10 and the high number at their situation, and this is the contradiction when I don't think if you said yesterday or today more or today is this is issue of go ahead. No, it's about the African country. When we discussed 3 years ago the aflatoxin in cereal, they are not happy because we put the maximum level too high. And yeah, so, and this is the contradiction. It's not a force.
I have something that I feel that is missing from the thinking. There is no benchmark dose, like a health-based benchmark value that I would like to have. And I would like to see how much public health gain I'm going to get. Even if my benchmark dose is not like, it's just for indication, because I want to see that so that I can compare how much public health gain I'm going to get for how much rejection. Because right now, all I'm seeing is relatively doubling the exposure, tripling the exposure, multiplying the exposure by 80. But I'm not seeing, versus what my benchmark.
But this is one of the problem. And when we try to calculation for those, we don't have the consumption data to do the.
So these are only max. Yeah, but even if I take
okay, we can do the calculation quickly. Yeah.
It's just to see it vis-a-vis, that benchmark dose. That's all.
Can you help me because I don't have in mind the total. What will be the limit here, or the benchmark dose for toxicology, for total aflatoxin?
Oh, this is aflatoxins?
Yeah, total aflatoxin.
Total aflatoxin.
Yeah,
we can have a look.
Yeah, I have just search the time that we can do. So you can do this exercise. We can say it's peanuts.
Yeah, but Moaz, if you have another consideration without it, I mean, I'm just telling you, for me, like to be able to make a response
exactly.
that I feel well constructed, I would like to see the comparison of how much, even again, it doesn't mean that I'm saying that there is no risk. I'm saying, relative risk. What is my margin of exposure? Basically, I would like to see what is my MOE. And where I'm moving my MOE to what extent. Exactly,
But, we need to do it with do it in average. We can do it in average.
With an average consumption, like we can have it. So
we can do it average consumption.
Yeah.
Because for me, there was.
Unless actually, because if I do it like that, I'm trying to think it's the same factor actually. Because if we fix all the other parameters, it's going to be the same factor.
It's the same factor.
So I'm trying actually to, I'm kind of answering my own question.
No, I think
or not.
I did it this way. At the end, I said it will be the same because it will be a multiplicative factor, not change.
Okay, so I think it's going to be the same factor. That's why.
No, it will be the same.
That's why. Okay.
But what we need is to have the baseline of what is the percentage of population exceeding.
Okay.
Just to have an idea about
that's
the base.
beyond. That's beyond. Yeah, we're not. That's kind of another type of estimation.
No, because when you look at the mean here, quite high.
Yes.
Yeah, this one is okay, perhaps.
Yeah.
But 1, 2, 1, 7, you still you are exposing your population to a certain. Yeah. So
okay.
So
these are PPBs, huh? The PPBs.
Yeah.
PPBs. Okay.
So when you look at finally, you can see that moving from because from 4 to 15,
Yeah,
the difference
it's 10%.
in any case, here you have a problem in markets. You have
Yeah.
to take ready of minimum 25% of your
Exactly.
production. You need to solve the problem for 25% of your production, not only by eliminating, but you need a reinforcement of
Codes of practice.
code
Yes.
of practice.
I think actually here, when I look at the data like this, my first temptation is to say, because between 10 and 15 right now, it's like the percentage of rejection based on that, it's
Nothing.
pretty much the same.
So you can
Like,
go to 10 easily.
I can go to 10. So I'm
Yeah.
going to take 10 because by doing 10, I might be getting better exposure reduction.
Yeah.
But again, I mean, I'm still concerned with a 29% rejection rate.
Yeah.
So of the achievability of that.
No, that's because, and here we need to go look at the data.
Yeah.
Geographical distribution. It is
Yeah.
and this is one of the problem when they submit the data. They don't want to give it country by country. They give it region by region. So all those issues. So we need to go back to the country and do better analysis and see it is distributed in the same region, the same area. Is it the same type of markets? Which type of markets? But that means if they are exporting, they don't have a problem. If you want, if they are exporting, perhaps they can go to 4 without any problem. But and they can explain that the contamination is mainly linked to storage in the local market rather than from the harvest and the storage when it is for the export. Just to have how much you to have it in a relative way. It's a relative, as you say, I multiply by 2 the exposure, or I multiply by 10 the exposure, or I multiply, so you multiply by a certain ratio the problem. Yeah, it's another way. It's my way of thinking. Yeah. Or you can look at it in another way. Here, if I do it in this way, where I will decrease by 80 folds the exposure if I move from here to here. Yeah. Yes. No, there's no bad question. Oh, you can have only good or bad answer.
I'm still wondering why there is this difference between the American and the European regarding the maximum limit of total aflatoxin.
It's not.
It was 20 for USA. It's still the total, and 4 for Europe.
No, because you have one continent who is importing, another is exporting. That's it.
So,
or another one. Yeah.
I have my other answer is also related to the way the ML is used as part of the toolbox of risk management. Okay, in the European Union, and this is my analysis, by the way. So I'm giving, this is my analysis. The organization that sets MLs is the European Commission, and it's not responsible for the conformity assessment and for the compliance verification and inspection, which remains the jurisdiction of member states. Now, at the EU, they try to level the playing field to make sure that countries with higher capacity are still having their consumers protected as countries with lower capacity than the others. Technically, if we want to apply, again, the Codex guidance about risk management, the way it would be is that you have to have your interventions in terms of prevention as much as possible, meaning inspection of the facilities, identifying of the issues. In fact, actually, one would argue even that when you set an ML, that's practically a maximum level that you are allowing to pollute. You know, you're giving that level. So of that, you give them a credit all the way to that level. So because in a single jurisdiction where the systems of inspections are unified, the guidance is unified, you can essentially have targets on the basis of exposure reduction by the interventions that you make at the national level because you unify those objectives. So you can achieve those by setting an ML that is still an indication. It doesn't mean that you do not capture things that are lower than the ML. So your action is not driven simply by the ML. The ML is simply a regulatory tool when you catch a non-compliant sample, but it doesn't mean that you would not catch something that is badly stored, something that is badly transported, anything that might be contributing. So that's why those jurisdictions, they set an ML, but they're not heavily relying on the ML. For jurisdictions where you have multiplicities of enforcement systems that are there, in order to protect everybody at the same level, I need to push these things down so that basically I minimize the potential for any disparity that I might have. And that's why, in fact, and this is my interpretation as well, that in the EU, you find that not only do they set an ML for the raw ingredient, but they will be ending up setting MLs for multiple, processed foods, because that's what people might be relying on, for verification. And again, that doesn't mean that the European consumer is better protected than the American consumer or the Canadian consumer. So that's why, it's really the way you're using the ML as part of a total risk management approach, and that's really key, because I can tell you, again, here I'm sharing experience in Canada. At some point, we were not setting an ML, but we were relying on activities of inspection and upgrades of the capacity. Mind you, again, for these type of products, Canada is a producer. So we were looking at really improving the capacity, and the actions were made on the basis of risk assessments that took into account not only the exposure of the consumer, but also the manufacturing practice, et cetera, et cetera. And sometimes action was taken for levels that were less than the ML that would have been set if we set one. You see? I mean, there are advantages and disadvantages to everything.
Yeah.
I guess the disadvantage is that you have, in fact, the disadvantage can be for industry. When you do not have an ML, you don't know to what level you are expected to comply.
And it is not predictable when they are exporting.
It's not predictable. Yeah, it makes it hard. So it makes it harder, So those are the type of things where, and that's why, I say sentinel levels. Sometimes sentinel levels you set for a product that is stored at a particular area, you can do a sampling plan. And if you see a level that is lower than the sentinel, it's okay. If you see a level that is higher than the sentinel, perhaps some action can be taken.
Yeah. And sometimes the industry, they want it. Yeah, they want it because they said, okay, we see give us the targets so we are sure that we are in compliance or non-compliance, and we can do our business without trouble. So this is a quick example. We don't have time because we have when we develop other workshop, we have a voting group with the full data, with different contexts, with different food type, and we have a discussion after that each one present the case, and we go to the reduction, because when you have, for example, for aflatoxin B, so we can calculate the number of cases. So you can look, because sometimes, if you move from 15 to 10, and you save one case per 100,000. Okay, so this is what you asked. And so of course it's hard after that to communicate that. One death, less death. but it's part of the discussion. This is part of the discussion at the cost of. So it's a trade-off after that. At the end, it's a trade-off between what you can achieve and, you know, and because you need to be careful that the maximum level will not be applied 100% because you are not inspecting all the batches. And you have the capacity of laboratory to be able to detect or not. So yeah, this is the maximum. So this is important to have it in mind. And after that is, for me, the same thing for microbiology. When you create microbiology criteria and sampling, we know that they are not enough, not because you put maximum level or you put microbiological criteria that you will solve the problem. But I think is a way to communicate with the industry and give them a target. Give them a target to achieve. So, and always the targets. I give always the example. If your son is not good in math, he has a score, let's say, he has 40% or 40%. You don't ask him tomorrow to have 90% score. So you do it by steps. Next, if you have 50 and so on. So, and all those countries, I don't understand their approach, and this is the complication. And here we see that there is the divide between the people that we have at the management with the people working on the industry, because they come with we want the best standards. We want it at 4, but they don't have any idea about the reality, how to achieve 4 or 10 or 5. And this is sometimes the difficult to see. I think they need to understand, and this we try to explain to them that they cannot come with very stringent to have the best regulation. No, having the best regulation, and you are not able to enforce the regulation. Yeah. No, it's not necessary.
Regulation that is not enforceable is a useless regulation.
Counterproductive.
Yes.
Yeah.
But I had a question about the initial setting of the 4, 10, 15. Why these values to work with? Exactly, but who exactly? But who decides about that? Oui, but who decides? Or relatively, let's say, to previous MLs that were set for other food matrices for the same contaminant?
Could have put 5.
But sometimes, so you have no. Sometimes in this CCCF, they start the discussion at 10, and there's no consensus. And the chair said, "Okay, I need a break." And after that, I said, "Okay, I discussed with the. I propose 15. Do you agree? Yes, no, yes. Let's move." Exactly that. We need to reach a consensus. Okay. And some people, they are not, oh, no, scandal, we cannot accept it. So, okay, that's the way. But if you want better, bring new data and show that you are, so if you bring new data and showing that you are able to reach better, so you can work for better maximum levels. And after that, this is the issue. The standards are at the global level. And you should represent all. So the standard will never represent the excellent situation. Yeah, the best, the benchmark. The standard is not the benchmark. So the benchmark of today can become the standard of tomorrow, but not the reverse way. Okay, so I think we can conclude here. You are tired. Yeah, you
I
deserve
think
it.
this is, yeah, this is, well,
can start.
Moez, I'm just gonna, in closing, truly on behalf of the team here and actually of everyone in the Codex community from the Arab region, and really very thankful. First, for the time that you dedicated to prepare these examples, for also the level of engagement and discussion. And I'm hoping again that this would be one opportunity amongst others that we can leverage, that we can build upon, so that we can continue to grow. I'm going to call it actually the food regulatory science expertise, because it's not only risk assessment, it's risk management, it's basically the mix, it's the entire food regulatory science component within this region. And we had the opportunity to discuss with Professor Hossein Masrouri, the Director General of the Oman Center for Food Safety and Quality. And in fact, on the margins of the organization of the Oman Food Safety Conference that is planned to be, I think, the third week of April of 2025, we will aim, based on, again, Dr. Masrouri's invitation, hopefully to organize some structured workshops, and perhaps, why not, even some in Arabic, some in English, to diversify this for the community and actually learn more from these experiences. So on behalf of everybody, truly, thank you so much. So thank you very much, Miles. And thank you to all our colleagues who have been staying with us. Actually, many of you have stayed for a number of hours with us online. So thank you for joining. I hope that you found this meeting and the discussions useful. And thanks to all who also contributed and participated in the third Global Food Regulatory Science Symposium. With this, I'm going to actually call it the conclusion of the third Global Food Regulatory Science Symposium, organized by the Global Food Regulatory Science Society, G-Force, in collaboration with all its partners, IOFAST, the Platform of Food Risk Analysis and Regulatory Excellence at Laval University in Quebec, Canada. And of course, without forgetting our generous hosts from the Dubai municipality and the Dubai International Food Safety Conference. Thanks to all of you, and see you very soon. Travel safe. Okay.